Okay, I think we're all probably here who's going to be here. So yeah, welcome to the Applied and Computational Math Seminar. We've got Giulia Celora today from UCL. Giulia started her career in academia in Italy before moving to Oxford. Yeah, she's going to tell us about modelling cancer. Over to you, Giulia.
Thank you. Thank you so much for inviting me. I'll give you a spoiler. This is my first seminar, a 1-hour seminar. So just bear with me. But as Tom said, I'm Giulia. I'm currently at UCL. I just joined UCL in October after finishing my PhD in Oxford. And today I'll be talking about some of the work I did during my PhD on the modeling of phenotypic and spatial heterogeneity in solid tumors. And this is work in collaboration with Ellen Byrne, who was my supervisor in Oxford, and Panos Kyriakidis, who is based at the University of Massachusetts Amherst. And I was lucky enough to meet before COVID hit, so we started a great collaborations. So why intra-tumour heterogeneity? Despite the advances and the research progress of the last decades in our understanding of cancer, the treatment outcomes are still very poor. And what people have realized is that sometimes failure of treatment is associated to intra-tumour heterogeneity. This is because our current treatment strategies are really good at killing the bulk of the tumor, the most of the tumor, but it's actually the few cells that are left behind that are usually resistant to tumor that then will eventually drive recurrence. And so our general goal and my general goal during the PhD was to use mathematical modeling to understand how the microenvironment, so basically the physical cues such as particularly oxygen availability that cells experience in the tumor shapes intra-tumour heterogeneity, because by understanding the forces that are driving intra-tumour heterogeneity, we can understand how to control them also in a clinical setting. So when we apply treatment. And so when you think about cancer cells, these are very complex entities. So they're kind of characterized by very different traits. Phenotype is basically like a trait. For the matter of this talk, I will focus specifically on one trait, which is stemness. It was more or less a decade ago when biologists realized that tumor, like normal tissues, have a hierarchical structure. So the idea is that type of cells with different developmental stages coexist in the same tumor. Here, I'm sorry if someone is colorblind, but the idea is that you can see some green and some red, and these are cells that are different. And so we have cancer stem cells. I'll now use another color scheme, but I decided that before finding this figure. But so we have cancer stem cells that are highly tumorigenic, so they are the ones that drive the formation of the tumor. And then we have differentiated cells. They usually consist as what the bulk of the tumor is made of, and these are highly proliferated cells, differentiated cells, and then eventually they have a finite life. So eventually they kind of burn out, become terminally differentiated, and they're just there waiting to die. And the idea is that there is this hierarchy such that there is only one developmental pathway. So they start with cancer, we start with cells being in a stem-like state, and they have this kind of infinite proliferation potential. They're kind of immortal. Eventually, they differentiate. Then they have finite lifespan, and then they become terminally differentiated. So they're just there basically hanging out to die. And this is kind of a normal pathways. But biologists have started realizing that the story is actually a little bit different. And this is really because of the advances in RNA sequencing technique, where they can actually look at these cells at the single cell level. And so here it's an image from some work from Baranowski, and they've taken some patient samples, they've done RNA sequencing and look at lots of genes. And then they have kind of look at the set of genes that for them was describing stem-like behavior, and then look at the expression level. So each basically histogram is a patient, and how much is the stem signature expressed. And as you can see, there is a large variability. So it's kind of difficult to describe the top figure with the kind of compartmentalized model, because it's not really what is a stem cells. You just like kind of drawing a certain threshold and say everything that is on the right is stem-like, but this is actually a good description. And so what has been realizing, what biologists have realized and mathematicians have been working on more recently, is the idea of that cells do not really like to live in boxes. This is just a model that we have, but rather they like to live along a spectrum, so a continuous spectrum. It's kind of the opposite as in physics. We moved from the discrete to the continuous now. And then there is another something else that the biologists have been realizing, and the idea is that there is not really a hierarchical structure. Cells are actually very plastic in the sense they can easily adapt to the condition they experience. So if you have cells that are experiencing high oxygen level, they're kind of happy, they follow a normal developmental pathway. The problem is that in the interior of the tumor, because of the imbalance between the oxygen supply and consumption, you have low oxygen level, and cells need oxygen to survive. So in the interior of the tumor, cells tend to de-differentiate, so go the opposite way, in order to survive as sort of protective strategy.
From a clinical point of view, do we, which is preferable? Do we want them stem-like or do we want them differentiated?
That's a great question. So I'll go back. So what they observe is that because of the current treatment strategies that we have, cancer stem cells are the most aggressive and the most resistant because they tend to be less proliferative than differentiated cells. So the idea is that when we talk about resistance, you need to think that based on our understanding of cancer, cancer wants to proliferate a lot. We can create treatments that target cells that proliferate a lot. But actually, we can't target those other cells that are there, are essential for the tumor development, but are poorly proliferative. But we do have other treatment strategies that can target these cells. They're just not yet in the clinic. So for this talk, I have divided the talk into two sections, and feel free to ask questions at any time. I will pause also in between the two sections. I don't want to go through everything. I just want to make it a next conversation so that it is also clear what I'm talking about. So in the first part, I will be talking about modeling the role of oxygen levels in shaping tumors development of phenotypic composition. And here I will start with a kind of warm-up problem where we look at the spatially homogeneous setting. And then in the second part, I will introduce spatial heterogeneity. And we can see how the introduction of space actually substantially changes the predictions of the model. And then in the second part, I would like to talk about how we can use what we had learned in the previous section to inform treatment strategies. So starting from an environmental homogeneous setting, this is, you can think about cells in a petri dish. So it's more of an experimental setting when you can control the oxygen level cells experience. And so our tumor is described by this phenotypic distribution that tells us at each moment in time how many cells have a certain phenotype S, where S is a continuous variable that goes in between S equals 0, completely stem-like, very aggressive, to S equal 1, terminally differentiated cells, poorly aggressive. And so our model describes how this phenotypic distribution evolves over time. And in the most general setting, what you can write down is a partial differential equation where you have a flux term, Js, that describes you how cells move along this phenotypic axis. So you can think that it really is transport, but in phenotypic space. And then, as everywhere in biology, we have a reaction term for proliferation and death. So as I was saying, we have proliferation and death that depends on the stem state of our cells. And because of the definition of terminally differentiated cells, we kind of assume that death is concentrated for cells that are around S equals 1. So this psi is basically a kernel function. Then one of the key features of this type of model that makes most of the mathematical complexity is that when we introduce competition for space, so the idea is that this tumor can grow up to a carrying capacity as normal. Here we normalize so the carrying capacity is 1. We introduce a null locality in the system, and this is a null locality in the phenotypic space. So here, this phi is actually an integral over the phenotypic space, and these are kind of more complex objects to deal with. And then from a biological point of view instead, the most interesting term is this phenotypic flux. How do we describe it? And the idea is that we introduce two terms. The first one is diffusion, and here is the idea that cells, there is some random fluctuation in the phenotype of cells. And then we have a second term instead, which is this advective term that kind of determines how the environment, so the oxygen level C, shapes the behavior of cells, drives the movement of cells along this axis. And of course, we have a no-flux condition because cells can only exist in this finite interval. And we have initial condition. So going back to the model a little bit more in the detail, as I said, there is a first term, the advection terms, that tells us basically captures environmental pressure. So in a high oxygen level, as we've seen before, cells tend to differentiate. So we have a positive velocity, so that cells tends to become thermodifferentiated. While in a low oxygen environment, cells tend to de-differentiate. So we have a negative velocity. And if you were to neglect proliferation at all, you can think, if you look at this equation, it's basically a particle in a potential. And so we can easily kind of intuitively understand what would happen on the long term. So in the first case, high oxygen level cells would accumulate around S=1 with some variability because of diffusion. So there is some thermal noise. In the contrary, if we have hypoxia, so low oxygen level, cells would accumulate around S=0 stem-like cells with a little bit of variability. But then we have this other term, which is proliferation with spatial competition, that actually captures natural selection. And so we have that in high oxygen environment, differentiated cells are the most proliferative. In a low oxygen environment, differentiated cells tends to lose their proliferation capacity because they can't adapt. And if we were to neglect the advection term, here we recover equations that have been studied a lot in the literature in the concept of adaptive dynamics, phenotypic models by other people that have done great work, specifically Lorenzi and Bertam. And that is that on the long term, we will have, because of natural selection, we would have that the fittest trait, so the one that proliferates the most, will eventually win. So in the first case, we would have like differentiated cells, a high abundance of differentiated cells because they are the most proliferative, and in the low oxygen case, we would have instead that cancer stem-like cells kind of win. But the key novelty of the work is that we introduce both. So we want to see how the competition in between these two mechanisms shapes the intra-tumour heterogeneity of the tumor in different environments. And so for the purpose of this talk, I will focus on stationary solution, and this is because we're interested in the long term, so how, independently of our initial condition, where the tumor is going to go, how its composition is going to be, and informed also by numerical simulation of the fluid dynamics, we kind of observe that the system always go towards a stationary solution. So we don't observe limit cycle, despite we can't prove it. So if you think about the stationary solution, these are determined by this stationary problem. And we have two types of solution. Phenotypic-homogeneous steady state, where basically there is no dependence on S. And here we have only one type of solution, which is the trivial, basically, solution. And we can use linear stability analysis to try and understand when extinction of the tumor would be possible. So when actually the tumor would go towards this trivial steady state. And then we also have phenotypic heterogeneous steady state, where therefore there is a dependence on the phenotypic axis. And here we can't really do much analytically because of the form of the equation, but we can use, we can compute numerically and both how these solutions look and also their stability by using something called numerical bifurcation analysis. And in particular, we use this bifurcation kit in Giulia, which is really good. I really recommend it if you want to study stationary solution. So starting from the trivial steady state, I won't go into the detail. You can do your standard linear stability analysis. And what is nice is that since you're linearizing around your trivial steady state, all of the complexity of the non-locality goes away. and leading order. And so you kind of get the Sturm-Liouville problem, for which we know a lot. So I won't go into the detail of that, but what does it tell us about the biology? So what we find for given biologically sensible parameter values is that in low oxygen level, the trivial solution is always unstable. So since it's linearly unstable, we're never going to observe its trivial solution. In high oxygen level, we can have that the trivial steady state is linearly stable for sufficiently strong environmental pressure. So if our advection velocity is sufficiently strong. So this tells us that elimination is possible, but remember, it's a linear stability analysis. So, you know, nonlinear perturbation might drive. So moving on to phenotypic heterogeneous steady state, we start with high oxygen environment. Remember, we have this proliferation profile, and we have environmental pressure. And as a bifurcation parameter for my analysis, I will use V+, which is the strength. How strong is environmental pressure compared to proliferation? So how environmental pressure compares to natural selection. And on the right, what I'm plotting is about our bifurcation diagram. So you do observe two branches. Here, when the branch is orange, is unstable. So if we start from this horizontal line, this is the trivial steady state. And we see that there is a change in stability through a transcritical bifurcation. So we do gather a little bit more of information. And this transcritical bifurcation happens where this non-trivial branch intersect with the trivial steady state. And what I'm plotting on the y-axis is you need to think that now we have a PD, so we are in an infinite dimensional space. And I'm projecting it by plotting the functional, which is the total mass of the tumor. So how many tumor cells we have. So if we are on the left, we basically have that the environmental pressure is subdominant. So we have natural selection dominates, and as expected, we have some variability, but differentiated cells are predominant in the tumor. On the contrary, on the opposite side, as we found in our linear stability analysis, environmental pressure dominates, and the only outcome could be extinction of the tumor. In between, what we find is that these two mechanisms are comparable. And so we find a profile phenotypic distribution, which is less variable than in this scenario, where cells kind of accumulate around S=1. But you can see a small bump of cells here, and these are still proliferative. So it's what allows the tumor to maintain basically and not go extinct. We can do the same. So again, using numerical bifurcation analysis in the case of low oxygen environment. So we have a different proliferation profile, and now environmental pressure drives cells towards stem-like state. And this is a little bit less interesting because, as we said before, the trivial steady state is always unstable, and cells tend to basically accumulate along S=0 stem-like state, because here natural predilection and environmental pressure basically drive the system in the same direction. And environmental pressure is simply decreasing basically variability. So to summarize this first warm-up section, we find that environmental pressure decreases tumor heterogeneity in general. Strong environmental pressure towards differentiation, so in the high oxygen case, can drive even the tumor to extinction. And we identify, but not prove, that there is a single stable stationary solution. So there is only one outcome, at least stationary solution, for any given choice of parameter values. And here, I think it's possible to prove it using the theory of positive semigroups, but it's more of a speculation because I haven't sit down and done the actual maths, but I think it's possible. So now the question is, we know what happens in a homogeneous environment. How does that translate to a setting in which we allow for spatial heterogeneity? Is there something new, or do we know everything by just simply looking at the in vitro setting? And so we start simple. So we neglect any variability in the y and z axis so that we can basically reduce our problem to a 1D setting, and we use x=0 as a basically symmetry axis. And I will present the equation all in non-dimensional form, where we non-dimensionalize time by the time scale of tumor growth, which is about 1 day, and x by the standard size of the tumor. It will just be complete. So our equation look very similar to what I showed you before. There are a little bit of tweaks to be made. First, we introduce movement of cells, simplest way possible. Cells are allowed to diffuse in the domain. And we assume that cells cannot leave our domain because it's a characteristic like patch of tissue, and so we have no flux at the boundary. We have an additional depth term. This is more biology, because when there is no oxygen, all cells will die independently of their basically phenotype. And then now, the way we describe our tumor is by a local phenotypic distribution. So at each location in space, at each given time, the local phenotypic distribution tells us how many cells have a certain phenotype. And furthermore, the other modification to the model is that now oxygen is not prescribed. Oxygen is supplied by the vessel, which is here thought to be located at 1. So we assume that oxygen diffuses into the tissue while being consumed by cell. And this introduces an additional null locality in the consumption term. So there is this phi here. And then we have that there is no flux of oxygen at x = 0 because it's a symmetry axis, and the concentration at 1 is fixed because it's where the vessel supply is. So now we have introduced much more complexity in the model. Before we had just 2 mechanisms competing, and we have them again also in the spatial setting. So we have environmental pressure, which now basically interpolates between the 2 behaviors we've seen before. We have natural selection by a proliferation, again mediated by oxygen. And then now we have also competition for nutrients, which is described by gamma, the consumption rate. And here we assume this is constant for all types of cells, and basically it goes to 0 where there is no oxygen. This prevents basically the solution to become negative. If there is no oxygen, cells are not consuming oxygen. And for the rest of the talk, I will define a tumor as being hypoxic if its oxygen level drop below this threshold. So basically, if there is the possibility, if there is a region in the tumor that basically favors the formation of cancer stem cells.
So, in your third bit, was there a gradient there?
Here?
Yeah.
Yes, it's very sharp. It's almost an heaviside function.
Okay.
Again, we focus on stationary solution. Dynamical simulation suggests that we always converge to stationary solution, not proven though. We have again phenotypic-homogeneous steady state. And we have the trivial steady state. The only one possible is the trivial steady state with the constant oxygen level equal to 1. Because there are no cells consuming oxygen. We can again do a linear stability analysis around the steady state. And what we find is that the result is the same as for the spatially homogeneous steady case with c = 1. And this is basically telling us that movement of cells doesn't drive anything interesting in this system. So elimination, as before, is possible if environmental pressure dominates, as before. Now we have also phenotypically heterogeneous steady states, and again we use computational approaches to compute their form and their stability. So if gamma naught is equal to 0, there is no consumption of oxygen. Oxygen is 1 everywhere. No surprise, we found the same bifurcation diagram as before, where we have a unique branch, which is a non-hypoxic tumor. So there is no dependence in space, as you can see that along the x, if you take a basically section along fixing x, and then the x is the same, and there is just some phenotypic variation. And this transcritical bifurcation doesn't move. If we increase gamma naught, something interesting happens. So we get our transcritical bifurcation at the same point as before, because we know the linear stability analysis doesn't depend on space. But now we have some new branches that are determined by the appearance of 2 saddle nodes, bifurcated points in red. So we again have our non-hypoxic tumor, which is basically spatially homogeneous and phenotypically heterogeneous. And then in the upward branch, we have this hypoxic tumor. So we have the formation of an hypoxic region. And you can see that now there is a spatial dependency. And we have that cells accumulate along S equals 1 in the high oxygen region, while in the low oxygen region, we still seeing the appearance of cancer stem-like cells. And so numerically, we can actually follow the location of these two bifurcation points. So now what I'm basically
What was your dashed line? What, are they stable or unstable?
Sorry, it's stable, it's green, stable. So these two are stable, right, and orange is unstable. The dotted line is just because it's, if you actually look at the stationary steady state, is different. So it's these heterogeneous, basically, spatially heterogeneous, right, okay, steady states. So it's to distinguish this top branch, stable branch from this one. So importantly, as you can see, if B plus is sufficiently large, we can have multi-stability. And so this is what we're studying now, because we have, as before, our bifurcation parameter is on the x-axis, B plus, but now we also have gamma naught on the y-axis, and we follow the location of our bifurcation point. So you can see that the two saddle nodes originate from a cusp point. And if you are on the left of this curve, basically, you have monostability. And you can have either a non-hypoxic tumor, so spatially homogeneous, especially heterogeneous tumor if you're like an hypoxic, spatially heterogeneous tumor around here. And if we are on the right of our transcritical bifurcation, we also have extinction. And this is for a range of biologically relevant parameters. Then we have multi-stability inside this red region, determined by two saddle node points. We can have coexistence of non-hypoxic or hypoxic tumor in this kind of narrow, dark green region. Instead, if we are on the right-hand side of our transcritical bifurcation, then we have multi-stability in between an hypoxic tumor, fully developed tumor, or extinction. And this is very interesting from, as we will see, a treatment strategy point of view. And so now the question is like, okay, we know that there are multiple fates for a tumor. And most of the scenario for this parameter regime are going to be in this hypoxic tumor or extinction scenario. So we will focus on parameters in this area. But now, what actually determines the fate of the tumor? That's what we are interested in. If we want to change of the tumor, the fate of the tumor, what are the features that determines it? And so we're gonna use dynamical simulation, and specifically we focus on one set of parameters. And so what? Start. So what you're gonna see here is a 2C set of simulation. All parameters are exactly the same. What we change is the initial condition, and particularly we change the initial mass of the tumor. So we have a smaller tumor on the left, a slightly larger tumor on the right. Everything else in the initial condition is the same. On the left, you're going to see the evolution of the total mass of cells, the total mass of the tumor. And on the right, you see the oxygen distribution on the top and the local phenotypic distribution on the bottom. And as we let the simulation evolve, you can see that initially, good tumors grow and cells start to differentiate. But in the smaller tumor, we see that there is a good oxygenation of the tumor. While on the right-hand side, we start seeing the formation of an hypoxic region that kind of introduces a spatial heterogeneity. And so eventually, from this point forward, we see that the trajectory of the two solutions starts diverging. One leads to the one that had initially a larger mass leads to a fully developed tumor, where the initially smaller tumor eventually goes extinct.
What's driving the extinction? I kind of missed that. Because there's no intervention here at the moment, is there? There's no clinical.
No, but you can. Exactly. So if you start when the two start diverging is when on one side you have the formation of an hypoxic region, and then hypoxic region cells tends to de-differentiate. So you have kind of because there is a niche, so a region of the environment that kind of drives the formation of stem-like cells, this remains like eventually accumulating the back of the tumor, but it can actually then move in the hypoxic region, in the normoxic region, and differentiate and grow. So there is a kind of
the cells in the core aren't
Exactly.
aren't used to the hypoxia that the region of the ring. There's lots of oxygen on the left, isn't there?
So lots of oxygen here on the right.
On the right.
On the right.
Oh, sorry, the left simulation.
On the left simulation, exactly. There is lots of oxygen.
Yeah.
And so the tumors tends to basically, the environment drives cells to differentiate. So there is no environmental cues that
Keep the
favors
cells there undifferentiated and
stem-like.
stem-like cells. So
Yeah,
they all
exactly.
just...
So everything is driven towards differentiation. And so there is no part of cells that can maintain the tumor.
So do I understand this? This is amazing, if I understand this right. That we should feed tumor cells to make them sort of happy and differentiate and give them all the nutrients.
That's something that they've done already. If you think about also, they were using I'm gonna get this wrong. But so they were used to put patients, before giving radiotherapy, they were used to put patients in high oxygen room to increase the oxygen, basically the pressure, because hypoxia, on the one hand, kills most of the tumor cells, but there is a high selection pressure for very aggressive phenotype, and so it's one of those riddles of cancers. What should kill cells actually favors the formation of the tumor. But I would say this is very theoretical work, so I wouldn't go and say this is what happens in a tumor. I would never. But so now,
on that question, then, the idea, has this been seen? The idea that small tumors, which are very well oxygenated, do disappear, or they break up?
I mean, the question is, would you even observe them? Because we don't really go and image tumors that don't go above a certain threshold. But you do have that smaller tumors that don't have a toxic region. If they form, they might be less. They are less aggressive usually. So we kind of observe this dependence of outcome on tumor mass, the initial composition of the tumor, and this is similar to what in ecology is known as a Lee effect. The fact that basically, if you have a small population, they might go extinct, but above a certain threshold of mass, the system would evolve towards an untrivial state. But now we have a phenotypically heterogeneous population, so we have an additional mechanism. So now what I'm doing, and I run a lot of simulation, and what I change is both the initial mass, but also the mean phenotype of the tumor. So basically the phenotypic distribution of the tumor. And if S naught is small towards 0, it means that I initially see a lot of cancer stem-like cells. And if it's a naught is large, I basically put in the system terminally differentiated cells. And we can see a good separation between simulation. And if I now plot on the left-hand side the outcome, the long-term behavior of the system, depending on M naught and S naught, what I find is that there is a nice trend here. I'm using a logarithmic scale, so it's very wavy. But we do see that there is a good separation between these two regimes. And in particular, we see that if the S naught is fixed, we do find a critical mass above which the cells, the tumor eventually will grow. And this is something actually you observe in an experiment, well, more or less an experiment. I'll see that in a minute. But so there is a Lee effect. But then what we see is that the critical mass does depend on the initial phenotype of cells. So if we have a small amount of stem-like cells initially, these are sufficient to drive the formation of the tumor. And this is what they observe also in vitro, because that's how they recognize actually stem-like cells, is that they can drive the formation of the tumor. Instead, even if we see the much larger amount of terminally differentiated cells, these will never be able to give rise to the tumor. And again, this is kind of nice because it's something that is standardly observed in the experiments. We didn't really input that in the model, but it kind of emerges. It's an emergent behavior. And so finishing on part 1, some of the key results. We have the tumors grow towards their carrying capacity, both in the spatially homogeneous and spatially heterogeneous scenarios. So it's this idea that we reach a steady state. The interplay between spatial and phenotypic heterogeneity can give rise to multi-stability. So something mathematically interesting, but also biologically interesting, because now we have that the tumor fate does depend both on its initial size and stemness composition. So on the one hand, we have that the fate of the tumor does depend on something that is not just parameter values, but it depends on its initial configuration. And specifically, we have we suggest that the initial size and how much stem cells are initially in the tumor can affect its fate. And so, okay, perfect. So in the last 10 minutes, I will switch to the last part, and so trying to tell you a little bit how we can use what we have learned in the first part of the talk to kind of inform treatment strategies, and specifically look at radiotherapy. So if you now look at radiotherapy treatment, we have an initial tumor that is fully developed, probably in a path of going towards a fully developed tumor. We apply treatment, but we apply treatment for a finite amount of time. So it's kind of, treatment is bringing us from one initial condition to a new initial condition, basically. And so the question now is like, is at the end of treatment, are we in an initial condition that leads us to a fully developed tumor, or that leads us to extinction? So we want to basically drive the system. Can we drive the system into the basing of abstraction of this elimination state? And so based on what we have just seen, there are two key features. We identified two key features of the system that determines its long-term fate. And these are tumor burden, so the amount of cells in the tumor, but also the fraction of stem-like cells. So the question now can be formulated in, how can we apply a treatment such that it leads us to this extinction, basically, regime, so that we both decrease the tumor burden, but we also decrease the fraction of stem-like cells. So we need stem-like cells. So in some way, we identify two metrics that are two predictors of our treatment outcome. And just to remind you, we have our local phenotypic distribution, which is the dependent variable in our model. If we integrate it both in space and phenotypic along the phenotypic axis, we obtain the tumor burden. So this is our first predictor. If then we take our local phenotypic distribution, integrate over space, and divide by the total mass, what we obtain basically is a probability. And that's a probability that by taking a random cells in our tumor at a certain time, its phenotype will be S. And then to identify what stem cells are, because here we go back to this kind of dichotomy between a continuous stemness axis and having to define some stem cells. And so we kind of impose a threshold and integrate the probability up to the threshold, and that gives us basically the fraction of cells that are stem-like. And so these are the two predictors that we propose can tell us about treatment outcomes. So now we want to test our predictor. We need to simulate radiotherapy. And here we use standard approaches. So radiotherapy, we assume that cells are killed instantaneously. This is an assumption, not the best one, but is a kind of standard in the literature. And we use standard models. I won't go into the details. The key feature is that at anytime we apply radiotherapy, cells die at the rate that depends both on their phenotype, and this is to capture the fact that stem-like cells are resistant to treatment, and this is observed, it's known. We capture also the effects of oxygen, because this is something else well known. So if cells are in an hypoxic region, the radiotherapy is less effective because it necessitates oxygen around to be effective. And then also depends on whether how much cells are proliferative, and this is something else that is usually taken in the literature. So now we have these two predictors. Our predictors evolve over time. So let's just look at the standard treatment protocol. We apply a 10 gray on Monday, and then we wait another week, and then apply 10 gray again. And we start our simulation with a fully developed tumor. And we want to see what happens. So we can pull over time our predictors. And so on the left, you can see the tumor burden. And as you see, every time we apply radiotherapy, there is a large decrease in tumor burden. And on the right-hand side, you can see is that the evolution of the fraction of stem-like cells in the tumor. And as you can expect, since cancer stem cells are radioresistant, we have an increase in the fraction of cancer stem cells whenever we apply radiotherapy. But then another feature that you can't really see here, but I'll tell you, is that as we apply radiotherapy, we decrease the number of cells in the tissue, and this leads to reoxygenation under the assumption that vasculature is not physically injured during treatment. And so if we have reoxygenation now, we know that the environment drives cells towards differentiation. So we do accumulate stem-like cells after radiotherapy, but then natural pressure naturally drives these cells to differentiate. So naturally plays in our favor in some sense. And so we have that in between radiotherapy session, we basically have that the fraction of stem-like cells decreases as long as there is oxygenation and competition. And this is telling us something interesting in the sense that I know it might be trivial, but sometimes when you look at how clinicians schedule treatment strategies, they don't really look at this. But the sensitivity of the tumor to radiotherapy does depend over time. So if you think about it, when in between the first and the second dose of radiotherapy, our treatment will be less effective because we have accumulated the resistance cell. So even if we apply the same fraction, we're going to be killing overall less cells. And so now we can think a little bit, how can we make our treatment more effective? Well, we've seen that in between doses, the fraction of stem-like cells decreases. So in between, after the first dose and before the second dose, we actually have that the sensitivity of the tumor decreases. So what if we wait a little bit more in giving our radiation? And so we can do this, and what I will be doing here, I will start with the same tumor. I will apply treatment, and now I'm going to change the amount of time I wait, delta tau, in between subsequent doses of radiotherapy. And I apply a certain amount of cycles. I let the system evolve over time for a long time, and I look at the final, at the final time point, I assess whether there has been recurrence. So I go back basically to my initial state, or if I can achieve tumor elimination. And now we can see that if we wait 7 days, kind of standard treatment protocol, even 80 gray of radiotherapy will not be sufficient to kill our tumor. Now, as you can imagine, giving radiotherapy is very toxic. So in some sense, we want to decrease how much radiotherapy we give. As we wait 10 days instead of 7, we start seeing that 80 gray is more effective. We start seeing that there is elimination. And if we wait 13 days, we can actually decrease that we can have elimination for just 70 gray. So we're kind of improving every time we increase slightly more how much we wait. But now if we wait too long, then we fail again. And this is because you can imagine that if you wait too long, you give the tumor the possibility of regrowing, and we have the formation of a toxic region. So we're going to have that we're basically too late. So there is this kind of nice non-linear effect in which we can find an optimal amount of time that we have to wait. Now I'm gonna say apply radiotherapy every 13 days. This is just to say maybe for a certain tumor it might be better to wait, and there might be an intermediate regime, an optimum that we can find for which we can be more effective. Not 13 days. It's going to be dependent on the tumor.
How many cycles did you go to get those rounds? Like how many kind of...
So here's 7 cycles. So 7 grays, 7 cycles. Yeah, sorry, because I put the end here. But so basically, it's total of 70 grays, 7 cycles. And not only now we have kind of got to a general principle by which we think we can kind of improve treatment outcome, but we also have some predictors. So what if we were to look at the end of treatment, look at our predictor, could we have said what happens on the long term? And so basically, as you can see, that if we wait 7 days, which we can think about as standard of care, we basically move vertically in this diagram. So we're decreasing a lot the tumor burden, but by doing so, we're not really affecting the fraction of cancer stem cells in the system. As we wait a little bit longer, we are less effective at decreasing tumor burden, which is usually what clinicians control and how they design radiotherapy cycles. But we kind of start moving also on the left. So we start affecting the fraction of cancer stem cells until we enter into the region where elimination is possible. But then, as we said before, if we wait too long, our tumor almost grows back to its initial size. We have the formation of hypoxic region, and so we have that. Also, the fraction of cancer stem cells at the end of treatment start increasing. So we kind of miss this beneficial effect of waiting. And so, looking back at our initial question, how does heterogeneity influence radiotherapy? The first effect that is well known is that to the mathematical community is that tumor sensitivity to radiotherapy, as many other treatments, is dynamic because treatment itself changes the tumor we're trying to treat. We can find a critical tumor burden, so a critical mass of the tumor above which, below which elimination is possible. So we need to control tumor burden as it's done. But actually, we also need to take into account the composition of our tumor. And when and how we can improve on standard of care, we propose some quantitative treatment and outcome predictors, so tumor burden as well as stem cells incidence in the tumor. And then there is this idea of treatment holidays as being beneficial. So the idea that the sooner is not always the better. So sometimes waiting can be beneficial. And this is seen before in other models. What is interesting is that we find also an optimum. So the idea that waiting too long also can be troublesome. And with that, I would like to thank you all for, even in a strike day, being here, also on St. Valentine's Day being here and listening to me for an hour. I would like to thank my collaborator, also Christos, who was in oncology at the time and helped us with the development of the model. These are some references, our two works on the Journal of Theoretical Biology, where we expand on this work, my research funding at the time, you again for listening, and this is just some contact in case you have any question or on anything, anytime. Thank you.
Excellent. Thank you very much. That's awesome. Any questions in the room?
So, really nice, very nice. Worried about what we differentiate to. So, do we have to be worried about you're differentiating? Your tumor disappears. I can see how you say you're removing from the stemness. That's the problem. If the stem cells stay, you can always keep using. I'm happy. I understand. We're good. Are they differentiating into more problematic cells like metastatic cells and then sort of floating around the body and creating problems elsewhere?
So here we go towards an higher level of complexity, and as most of them know, biology is very complex, and I'm not an expert. But what they observe actually is that usually they need to have an EMT that basically allows you to move from epithelia to mesenchymal that are usually metastatic. You actually go through a stem-like state. So you need to revert. And usually they are this intermediate EMT that they observe. They think it's in a stem-like state.
So by doing this, driving away from
differentiation is more like I basically lose this infinite proliferation capacity that I have, and I become less. I lose the ability of basically adapting to environment around me. So that's as long as our stemness definition goes. Then you can have different type of differentiated cells because the tumor is, as any tissue, very complex. So yes, you're right. There is much more, and there are like many other, as I said, many other traits that define cancer cells. So I'm not gonna say go and apply this to the clinic. It's just finding some general principle by following what a biologist formulate in words using maths. What are our conclusions? And then we can start thinking for sure, how can we test this, first of all, in a Petri dish, even doesn't make any sense at all. But at least it's like, you know, we have all this amount of biological literature with reviews on stem cells and these ideas of how they behave. Let's put that into a mathematical model and then see like what interesting, non-trivial findings we have. And I think like most of all in this special case here, we do see a rise in some interesting feature.
That's really good, thank you.
Yeah, so it was great talk, really. So thank you for that. When you were talking about the bifurcation diagrams in the heterogeneous environments, you said that it really didn't depend on the spatial aspect, the idea of space, it was more the environmental pressures. Do you think that that maybe is kind of a component of the symmetry induced in the system with the 1D analysis? Would this kind of, would you expect that to stay there if you had more of a complex geometry you're kind of playing with?
So I think, so this is a really question. First of all, I'm fixing here the diffusion coefficient to a certain parameter. So diffusion coefficient would kind of move around these boundaries basically, and how much distinction there is in the phenotypic state of the two basically region. That would affect the stability, but we kind of have, most of all for solid tumor, a kind of understanding on how fast more the order of magnitude is the right one, and it kind of doesn't change too much. So the idea is that I'm fixing many other parameters. What I tried to do actually was the way in which we introduce movement, for example, is a simple diffusion model, which can be poorly, a poor description of what is actually going on, but it's the simplest way in which you can introduce space. You can think about maybe more advanced models. I tried with the standard one-phase moving boundary problem. So if you have like a Greenspan type of model, but with phenotypic heterogeneity, and basically diffusion disappears. And overall, it's more or less the same. So we do find, I think what is key is that how this diagram more or less look would change, but the fact that we do observe this stability between extinction and the non-trivial steady state does exist, and is kind of pretty strong. So it's more like where the fact that there is this transcritical bifurcation is kind of the key feature, and the fact that it's this other branch. But the details of it will be dependent on parameter values, how you introduce space. And this is actually an interesting question, I guess, also in general, from the point of view of we have so many models. We do introduce different way of modeling, like effect of space, consumption, movement. We're trying to capture the same mechanism, but in many different ways. Does it matter what kind of equation we input, or is there something that is robust? What are our robust predictions? And I think the fact that this is pretty robust is quite interesting. But there is a lot to explore.
We'll have plenty of time for more questions and discussion. Yeah, thank you again.
I said to Robin, Tuesday afternoon because